IBD globally affected more than 20 million patients.
And the problem is that you've got tiny areas of damage in the intestine. So we thought about identifying a reparative compound, providing for epithelial mucosal barrier repair and closing those tiny damaged areas.
So I've never seen any compounds works in all the models.
That's why it went through peer review in Nature Biotechnology.
Last year, In Silico Medicine published a study in Nature Biotechnology highlighting its AI-designed dual PHD1/2 inhibitor, ISM 5411, which represents a major breakthrough in the treatment of inflammatory bowel disease. Developed using the generative chemistry platform Chemistry 42, the compound features an innovative gut-restricted pharmacokinetic design that minimizes systemic exposure risks. ISM 5411 demonstrates significantly higher inhibitory activity against PHD1/2 compared to existing candidates.
So Alice, congratulations on the publication of the Nature Biotechnology. And more importantly, it's not only about the paper, it's about the science behind the target and also the program, the compound, right? So we found a very good, safe molecule to treat IBD with a novel MOA comparing to all the existing therapies for IBD.
Very, very happy to announce the publication of our breakthrough paper in Nature Biotechnology. And while we were in review, we actually have completed the phase 1 study. So that is a pretty dramatic event because now we have several clinical trials out of generative AI completed with safety, and in the case of the TNIC inhibitor, we have efficacy. Very, very strong signal, dose-dependent. Here, we're talking about IBD, inflammatory bowel disease. It's a very large market. A lot of pharmaceutical companies are now rushing into the IBD market with different compounds. Most of them have anti-inflammatory properties, targeting directly inflammation. Some of them are addressing the microbiome, so different directions. However, they are not solving the problem. And the problem is that you've got tiny areas of damage in the intestine, and those tiny damaged areas are creating the source of inflammations. So we thought about identifying a reparative, regenerative compound that would solve a problem. So providing for epithelial mucosal barrier repair and closing those tiny damaged areas. And so far, attempts to do that have failed.
Also for this target, we really have to thank our Pandomics AI platform, right? So we compare the patient's data with the healthy people's data, and in all the analysis, PHD1/2 appears to be the target that the AI selected for us for the IBD, for the barrier protection, and also the HIF1 alpha. So everybody knows that HIF1 alpha is regulated by PHD1/2, right? So we did see some strong correlation between this target, PHD1/2, and the disease, IBD.
Yeah, so that is a very strong evidence, again, that AI can pick the right target. Originally, when you come up with a list of targets from AI, what you need to do is you need to test them in a variety of experimental models. So you first go into cells, you go into animals, before you go into humans. Before we did that, we needed to achieve a high dose for IBD. So to do that, we wanted to go after gut-restricted molecule. to ensure that it works on the gut and doesn't go into the blood, in addition to being reparative and having high combination potential with existing anti-inflammatory drugs and different other therapeutic modalities for IBD. And we utilized Chemistry 42, another engine that in silico has developed to design molecules with gut-restricted properties. So you don't get them into the blood. So it's become safe by design, and you just excrete the molecule after it's done its job.
Yeah, and I think, how we use AI to design the gut-restricted molecule is one of the highlights for this Nature Biotechnology paper, right? So the editor specifically asked us the question, how you use your AI to design gut-restricted molecules, right? So basically, just for the gut restriction, there's no rules on how we can, design specific gut-restricted molecules. But what we did is we use our AI to predict a molecule's solubility, permeability, clearance, right? So in order for the molecule to be gut-restricted, your molecule cannot be too soluble, but it cannot be too insoluble, right? If its permeability is excellent, it will pass the gut barrier very easily. But it cannot be with extremely low permeability, otherwise it cannot get into the cell, right? And also the metabolic stability need to be reasonable, right? So we use our AI, Chemistry 42, to predict those profiles of each molecules, and we need a balanced profile. And once we have all the predictions there, we selected the top molecules that are predicted by our Chemistry 42 that can be gut-restricted, and we synthesize those molecules and test it. Fortunately, we got this molecule very quickly, and when we test it in the mouse, it shows gut restriction, and we test it in the rat, in the dog, it always shows the gut restriction. So that's one of the highlights of our technology, using AI to design gut-restricted molecules. Maybe in future, if we have to, for other programs, we have to limit the drug into the gut, we can use the same strategy, use the same technology to do that.
So yeah, if you are watching and if you are in Big Pharma and if you're struggling with achieving gut restriction, please do give us a call.
Yeah.
We are always welcoming pharma collaborations to achieve specific objectives in terms of molecular design or picking the right target, designing the right molecule. And here is a very good, clear example of how it was done. It was also done extremely quickly, so with extreme efficiency, thanks to Dr. Ran's genius and the team of expert scientists. Again, the milestone was achieved very quickly. I think it was below 12 months, right?
Yeah, because the gut-restricted molecules have a lot of advantages over the molecules that have the blood exposure. Because the molecule, if it's gut-restricted, it can really have very safe profile. Even at that dose, 1,000 mg per kg per day dose in a rat and also in a dog, we didn't see any side effects. And even in human, in our phase 1 studies, we dose the human up to 1 gram per day in a multiple ascending dose, and we didn't see serious side effects. So the molecule is really safe so far.
But you see amazing efficacy.
Right.
And in the paper, we've reported, of course, multiple preclinical experiments, but the highlight were the 3 experimental models of ulcerative colitis, inflammatory bowel disease, where the molecule performed extremely well. So if you compare the guts of those experimental animals with and without the drug, you want to be the mouse on the drug.
We use a lot of animal models to test our compounds. So that include the TNBS model, DSS model, oxazolone model, and also T-cell transfer model. So basically, we use the majority of the model available in the CRO to test our compounds. So to our surprise, the compound worked in every model. So I've never seen any compounds works in all the models, right? So I work in S1P program before. I know the JAK program, so they work in some model, but not all the models, right? Some compound work on TNBS, some compound work on oxazolone, but I never seen that one compound can work on all the models we tested. And also because the molecule is a barrier protection, has a barrier protection function, it can combine with other anti-inflammation compounds, right? So we also try those combination models in our preclinical studies, and we saw the synergy. For example, it can show some synergy with the TNF alpha antibody, so that you can combine the anti-inflammation and also the barrier protection. So it have a huge disease expansion possibility.
Potential, yeah.
Potential. Yeah.
So the indication expansion potential of this molecule is enormous, because barrier repair, we actually need that everywhere.
Yeah.
Barrier repair is one of the kind of central mechanisms by which you can possibly extend lifespan and improve overall well-being, because during aging, the barrier function is diminished in both the gut and also in the brain, and even our skin gets worse, right? So barriers are very important for preserving our life ecosystem, because we're all made of cells. And achieving efficient barrier repair provides you with massive indication expansion. You can go into dermatology and many other disease areas. But of course, our primary objective is to go into IBD, get a very clean clinical readout, and preferably combine it with several drugs. The feedback on this molecule has been extremely positive, with no negative marks, so to speak. And that's why it went through peer review in Nature Biotechnology. And we are very happy that it went through and is getting published, because now you can read about all of those experiments, see how they perform, how the molecule performed, and learn, first of all, about how the molecule works, how the target works, formulate additional hypotheses, and learn how to combine this molecule with other molecules for even more efficient barrier repair, try to do indication expansion. We're extremely collaborative. And what's most important is that now the phase 1 is complete in Australia and China now as well. So that 2 phase 1s complete. By the time we got the paper accepted, the China trial was still ongoing. But these are the actual pills that were used in the study. So you can see the size of the tablet. So it's pretty big. So yeah, you basically excrete it, right? It doesn't get into the blood. So you need to give a lot of it to get to high dose. This is 200 milligram tablets, so you actually need to take several to get to a gram. And it's one of the, another AI-generated compounds that were designed or discovered utilizing AI, designed utilizing AI. And of course, there is massive amount of human work involved. So human ingenuity at this point in time cannot be fully replaced by AI. That is why AI is just a tool in our hands and in the hands of the others, because now you can buy the same software and acquire the same capabilities. So all of these software tools that were used for the study are commercially available. And we're very, very happy to be empowering the community to achieve similar results. So if you are interested in doing more testing on this, or making a home for this particular product, please do give our business development team a call, and make sure to read the Nature Biotechnology paper, because it's a huge highlight. It's our fourth.
Yeah. So it's the second this year, right? And the fourth in total. And also IBD globally affected more than 20 million patients, right? So we finished the phase 1, and we are going to start treating patients next year, in year 2025. So hopefully we can share with the audience, our clients, more exciting data when we see some efficacy signal in the patients.